Bismillah ar-Rahman ar-Rahim. Today we are going to start our course in biology for standardized test ACT 2 in Egypt or ACT 2 in some countries. Let's have a quick look on the curriculum of biology. These are the courses for 2024-2025. As you see, this is the calendar, August, September up to July. 2025. Here are the dates of the EACT2 sessions and EACT1. EACT will be on October, on December, then Jan, March, May, July. How many sessions of EACT are available for us? 1, 2, 3, 4, 5, 6 sessions from now, right? For students who are planning to run the ACT test, here are the calendar and the deadline dates, and SAT1, of course, in other countries. Biology ACT have 8 main topics. Cellular and molecular biology, it versus about 20 to 25% of the course. Heredity and molecular genetics from 20 to 22 or 23% of the course. Diversity and evolution. We can put them in one topic or 2 topics, but I prefer to put them in 2 topics. This is the topics for Kabbany Academy. Kabbany Academy is a school online, www.kabacademy.net. Diversity of living organism, about 10%. Evolution, about 10%. Ecology, more than that, I think, from 10 to 15. Plants and plants biology, less, about 5%. Human and animal biology from 10 to 15%. Animal behavior, only 1 question, only versus more than 2, 3, 4 questions, up to 4% of the curriculum. Cellular and molecular biology describes introduction to biology, biochemistry, enzyme, cell structure and function, homeostasis, cell transport, photosynthesis, cellular respiration, and cell division, and so on. These are the subtopics of the curriculum. Types of courses, full year course. This is very, very suitable for students in grade 10 and 11. And full year course targets 800 out of 800 for ECT. Full semester course will end in Jan. Okay, this is preparation for Jan test, right? Half semester course, this is very suitable for half semester course, this is suitable for December. Next trial course, it is like quarter test, like quarter, yeah. It's usually 8 weeks. Look, full year course, this is very suitable for full year course, this is suitable for grade 10 and 11, full semester course, half semester course, coming trial course, panic course, if you are running the exam and you're coming the day, the night before the exam and tells me, save me, I'm going to join the test tomorrow, I can help you too. this Hispanic course for 1 to 3 weeks, and peer sessions for some students who have special situations. Okay. Full year course targets 800. Full semester course for good students up to 700 out of 800. Half semester course, you are targeting 600 to 650. Coming trial course, don't think you can get more than 550 to 60 to 600. Panic course, here I think it is hard to get 600. Panic course, it depends. It depends. And peer teaching, it depends. Okay. And these are the full year course curriculum distribution. This is chapter 1, lesson 1, 2, 3, 4, 5, 6, every one lesson per 1 week. Full year course, 1 lesson, 1 week. Full semester course, half this. December course, look, I usually combine these courses together. Full semester course and December. Only the difference in full semester course in Jan preparation course, only you have more time for revision and exams. And in panic course or next trial course, usually we skip some topics like plants and plant biology, like half of diversity of living organisms. We skip these topics long and worse very little. Here we'll work on 5% only, and this we are going here work on 90% of the topics. Okay. Anyhow, this is the course description, and let's start with introduction to biology. Cellular and molecular biology for standardized test. This is the course description. These are the 8 chapters and the types of courses. And these details of the lessons that belong to each chapter. Okay. These are the curriculum distribution done at Cap Academy. And this is the calendar for 2024, 2025, and types of courses, right? Let's start the lesson. Life functions. What are characteristics of living organisms? What are characteristics of living organisms? Okay. Life functions. All living organisms, whether unicellular or multicellular, carry out certain function processes, life functions or life processes like ingestion, digestion, respiration, transport, regulation. Okay. What's ingestion? Ingestion is intake of food, intake of nutrients, intake of food material. In animals, of course, you swallow. Okay, like swallowing. Animals, you swallow. In plants, we take nutrients across the roots, right? Unicellular organisms, either by diffusion, active transport, or endocytosis. And all of this will be discussed in details. This English intake of food. Here we're talking for unicellular and multicellular, all types of living organism. Digestion. Digestion is the breakdown of large food molecules into small food molecules, soluble in water and absorbable by small intestine for human. Okay? Here, small, large molecules to small molecules to be easily absorbed. This is called chemical digestion or hydrolysis. Hydrolysis. Lysis means breakdown. Hydro means water. This breakdown of large molecules using water. There is another mechanical digestion. This is chemical digestion. Here, mechanical digestion. Large pieces. You swallow a piece of cake or something like this. Chewing, example, chewing. Into small pieces. Okay? From large pieces into small pieces. This will give more surface for enzymes to act on them, right? To increase surface area. Mechanical digestion, the aim is to break down large pieces into small pieces or to make better mixing of chemicals or the enzymes with the food molecules. Here, increase surface area for contact with the enzymes. Remember when you put a cube of sugar in a glass of tea and sugar powder in another glass of tea, the sugar powder will dissolve more easy because small particles will have more surface area to act with water to dissolve. Enzymatic breakdown, here is talking about chemical digestion. Enzymatic breakdown, this is chemical digestion, called hydrolysis of food. So it's small enough to be absorbed and assimilated. What is meant by assimilated? Assimilation means to be respired. This means to be burned inside the body to release energy or transformed into other molecules necessary for the body. Being burned, being broken down, these molecules absorbed to the body, being broken down or changed to other molecules. This is called assimilation. Assimilation is a character of life. This means dealing with molecules inside the body. Respiration. Respiration is not breathing. Breathing is another thing. Breathing, in-breathing, inhalation, out, exhalation, inhalation, exhalation, taking air in, taking, this is mechanical process. But respiration, burning of food to release energy, burning of food to release energy. Inside every living cell, using enzymes. This is called respiration. Exactly like you put fuel into the car and have a long trip to Alex, and the fuel is finished, consumed. Where has it gone? Where has it been? When has it gone? This fuel has been burned to give energy to the motor. to work inside every living cell. This is metabolic process that produce energy. Energy in biology, in living cells, called ATP, a molecule we are going to study in detail. ATP, adenosine triphosphate. Transport all living organisms, plants, animals, microorganisms, unicellular organisms, are able to distribute molecules from one part to another, from one part of the cell to another part of the cell, or from one cell to another cell. Transport. So you have transport system in your body, transport system. Sometimes you call it circulatory system, the heart, the blood, and blood vessels. And inside the cell, there is something called cytoplasmic streaming moves like this. Cytoplasm moves around the nucleus. Regulation. Living organisms are able to regulate themselves to keep their internal environment constant, stable. This is called homeostasis. Homeostasis is a very important term in biology. Homeo means the same. Homeo means the same. Stasis means stable. Homeostasis, keeping internal environment constant. Keeping internal environment stable. For what? For temperature. Your body works best at 37 degrees centigrade. This is your body temperature. Above this or below this, lowers the or affects the function of body function worked by enzymes. pH, it is a measure of acidity and alkalinity in your body. The pH of blood is 7.4. Okay. Above 7.8, you are liable to death. Below 6.8, you are liable to death. This is acidic and this is alkaline. Here, coma, is in coma. And water content. Of course, you heard of dehydration. Water content. Okay? If the body loses water, okay, the body ceases to make sweating and you search for water. If there is overhydration of cells like IV, if you put IV fluid in hospital and make over intake of fluids, this is harmful. The body may kill the patient. Regulation is keeping internal environment constant, and this is a very common question, very important term. Synthesis, exactly like the body, your body is able to take large molecules and break them down to small molecules. Okay. It's able to make large molecules from small molecules. Example protein is formed of amino acids. You have 21 amino acids in your body, different amino acids. Of course, you have many, you have millions of amino acids, but amino acids in formation of protein, only 21. Protein amino acids, only 21. Okay? Very nice. The sequence of amino acid number 1, number 2, number 5, number 7. This is the arrangement of amino acids, say in beef meat. But your body meat is different from beef meat. So it catabolizes, catabolism, breaking down food digest protein into amino acids, then rearrange them to make your own protein. Rearrange. Number 5, number 20, number 21, number 15, and so on. The sequence of amino acids. So your body flesh is different from chicken flesh, different from fish flesh, different from beef flesh, right? Synthesis is combining small molecules or substances into larger, more complex one for body functions. Excretion, removal of metabolic wastes. Okay, as you burn food on the oven, it produces smoke, carbon dioxide, and water. Also, burning food inside the cells gives metabolic wastes. Carbohydrates, when burned, it gives only carbon dioxide and water. Protein, when it burns inside the body, gives the carbon dioxide and water. And ammonia, NH3. Ammonia should be changed into urea, uric acid, come to excreted through skin, sweat, and the kidney, urine. Proteins, fats, usually incomplete combustion, give carbon dioxide, water, and some acids, ketone bodies. It's harmful to the body. So it's very, very easier to burn carbohydrates, especially glucose. It's a common question. A quick and fast source of energy is glucose. Is a quick and fast source of energy in your body. Egestion, what's egestion? This excretion and this egestion. Right? Egestion, if you swallow food and a very, very small stone, the stone passes undigested until it comes out through the anus. This is called egestion, not excretion. Removal of undigested food. In the same way, this is an organic molecule, and this is amoeba. Amoeba comes here to engulf the food by something called endocytosis. Then the organic matter is pushed inside the amoeba. Here, digestive enzymes through lysosomes join them, digest it, and into smaller molecules absorb useful substances like glucose, amino acid, and then egest undigested food. That remains. Here ingestion, here egestion. Ingestion, and here egestion. Okay, stool coming from the anus is egestion. Most of contents of stool are egestion, undigested food. Your knowledge, a cow eating 100 kilograms of grass egests 40 kilograms. Okay, 50% are burned. 50% are burned through respiration to give energy, and only 10% is changed into meat, milk, and other, right? Digestion is the removal or passage of undigested wastes. No, not undigested waste, not undigested food, right? Reproduction, this is the ability of the living organism to reproduce, to produce offspring of the same kind. Okay? offspring of the same kind. This is reproduction. When we marry and reproduce, some unicellular organisms reproduce asexually. Okay? And there is a lot to be discussed in reproduction. Irritability. Irritability is the ability of the living organism to feel changes in the environment. This means to detect stimuli. Detect stimuli. What are stimuli? These are changes either in the external environment, like it's hot, it's cold. Somebody insulted me, somebody beated me, and respond to them. Every living organism, whether multicellular or unicellular on Earth, has irritability, detect stimuli and respond to them. Right? What are stimuli? Anything, any change in the environment, the outside your body, inside your body, that stimulates you to respond. And many words to be said. Movement. All living organisms are able to move. Okay. Amoeba moves. Paramecium moves by cilia. Here, amoeba by zoodopods. Zoodo means line. Pods means legs, pseudopods, amoeba. Paramecia by cilia. Some bacteria by flagella. Plants. Plants move some parts of the body, like leaves move, right? Like phototropism. The plant leaves move towards light. Slow, of course, slow movement. But it moves part of the body. Animals move. Walk, run, right? This is called animals. This is a horse, okay? Suppose it's a horse. It runs, right? This is called locomotion for animals or animal-like cells, like amoeba. Metabolism, what's meant by metabolism? Metabolism. All chemical reactions inside the body are called the metabolism. All chemical, sum total of all life functions, all chemical reaction. Life is a chemical phenomenon. Life is a chemical phenomenon. Life is chemical process. Everything you make, even vision, even memory, this is chemistry. Metabolism consists of anabolism. Ana in Latin means up. Anabolism is building up processes. And catabolism. Kata means the breakdown. Catabolism, breakdown. Ana and the kata. Anabolism and the catabolism together for metabolism. Respiration, burning of food. Of course, this is catabolism. Photosynthesis. This is anabolism. Growth, anabolism, right? Digestion, catabolism, and so on. These are the characters of living, functions of life or characteristics of living organisms: ingestion, digestion, respiration, transport, regulation, synthesis, excretion, ingestion, reproduction, irritability, movement, and metabolism. This is topic number 1. Topic number 2, measurement, like metric system, measurement. metric system and measuring. We use the meter, small m, okay? Meter, small m. This is the measuring of distance or length, okay? Here you need to know some prefixes. The prefix milli means 10 power negative 3, 1,000th. Okay? Micro, 10 to the power negative 6, 1,000,000th. Nano, okay, 10 to the power negative 9. Millimeter, this is the small dash on your roller. You know, your roller is like this. Your roller is like this here. This is centimeter and this centimeter. Centi means 1/100. Hundredth centimeter. And these small dots, millimeters. Of course, 1 centimeter equals 10 millimeters, right? So millimeter, micrometer, nanometer. Your eye is able to see, to visualize 10th of millimeter. The light microscope is liable to visualize, to make you see 1/10 micrometer. Worth notice, micrometer like amoeba, like bacteria. Amoeba 10 micrometer. Micrometer like bacteria, like mitochondria and others. Nanometer, the electron microscope, 10th of nanometer. This means 10 to the power negative 10 of meter. This is called angstrom. Pico meter, 10 to the power negative 12. There is femto. Okay, pico, femto. Okay, look here. The electron microscope can make you see angstrom. And the nanometer, we use measurements of atoms as a whole. Picometer parts, subatomic particles like proton, the neutron. Picometer, okay? These are the lengths. In our work, we will work on, usually in biology, micrometer, 10 to the power negative 6 of meter. Nanometer, 10 to the power negative 9 meters. Suppose amoeba 50 micrometer. Change it into meter. Change it into millimeter. So 50 micrometer. What do you want to change it? Change it into millimeter, okay? X millimeter, right? Here. 1 millimeter equal 1,000 micrometer. You jump from meter to millimeter to micrometer to nanometer to picometer. Every jump 10 to the power 3, 1,000 times. A meter is 1,000 times bigger than millimeter. Millimeter is 1,000 times bigger than micro, 1,000 times bigger than nano and pico. Okay. فهمتوا قعدتوا انسوا. This like this, okay, equal 50 divided 1000 millimeter. Okay. Mass. The international unit of mass is kilogram. Don't write K capital. No. This is Kelvin. K small. Kilogram, right? A kilo, the word kilo means 1,000. Kilogram means 1,000 gram. Kilowatt, 1,000 watt. Kilovolt, 1,000 watt. Kilo ampere, 1,000. Kilo viewers, 1,000 viewers. K means 1,000. Okay, from kilogram to gram, 1,000. To milligram, 1,000. Okay. To microgram, 1,000. To nanogram, 1,000. To picogram, femtogram to attogram, and so on. When you have 5 kilogram, want to change it into gram, make like this, into gram, right here, kilogram, and make them one, as if you want. To make this one, say 1,000 gram. Now your work is in grams. You need to make milligram. Okay, no problem. Milligram over gram. And make this one. Okay, to make this one, a gram, 1,000 milligram. Change into microgram and so on. Continue this conversion factor. You are familiar with milligram in drugs. You are familiar with kilogram when you buy potatoes, tomatoes. You are familiar with grams when you buy gold. Okay? You are familiar with milligram when you take antibiotics. Penicillin, 500 milligram every 6 hours, or 1 gram per day. Augmentin, streptomycin, kanamycin. Okay? Micrograms, you are not familiar with it in biology. In chemistry, yes. Yes. Okay? Microgram. A microgram, a millionth of gram. Gram, milligram, this is 1,000. Microgram, another 1,000, right? Volume. When discuss volume, look here. You are familiar with centimeter. When you make a small cube of centimeter, one centimeter, 1 centimeter makes centimeter cube. Okay, centimeter cube like syringe. I have syringe. This is syringe, 2 centimeter cube, right? 2 centimeter cube. 1,000 centimeter cube makes what? Okay, look here. If you pick 10 centimeter and 10 centimeter length, width, and height, okay, 10 centimeter, 10 centimeter, centimeter, it makes 1,000 centimeter cube, 10 cube, right? 1,000 centimeter cube, 500 times like this thing. So the word thousands means kilo or milli, right? So 1 centimeter equal 10 to the power negative 3 liter. This is liter. Liter of milk, liter of drink, liter of anything, right? So centimeter cube equal milliliter equal 1 to the power negative 3 liter. And liter is 10, 10, 10, right? A bottle of infusion at hospital, in the hospital, here 500 milliliter or centimeter cube, right? Okay, IV drip, right? Yes, a liter of milk, a liter of water, okay, a liter of drink, all of these liter, milliliter. When you get syringe, 5, 1, 2, 3, 4, 5, 5 milliliter, 5 centimeter cube or 5 milliliter, M small, and liter versus 1,000 of centimeter cube. Okay? These are the SI units in biology, chemistry, and physics. These are some derived units like area, density, mass per unit volume. Okay, some prefixes you may find them. If base unit is 1, DC means a tenth, like decimeter. A ruler, small ruler, 20 centimeter long, this is 2 decimeter. So sometimes they say that the liter, say a liter, this is a bottle drink or something, liter, equal decimeter cube, 10 centimeter decimeter cube, okay? 10 power 3 centimeter cube, decimeter cube. Because a liter, 10 times 10 times 10, length, width, height, 10, 10, 10, right? Centi, hundredths. Milli, thousands. Micro, nano, pico, okay? So, from centi, from milli to micro, this is important in biology. From micro to nano, this is important in chemistry, atomic chemistry, right? Micro, a thousandth of milli. Milli, a thousandth of gram, and so on. If you go up, deca means 10, hect 2 means 100, kilo, mega, giga, tera, and so on. Topic number 3 in introduction is accuracy and precision. I am a physician. Originally, I used to be a physician, pediatrician. And I have 5 assistants. I have 3 nurses in my clinic. I give them a little baby. I know, and I'm sure this little baby is 10 kilograms. I told them, weigh the baby. Okay. Miss A told me that the baby is 10.1. Fatima told, no, it is 10. Salma told me that no, it is 9.9. All of them are different in the last digit. They are measuring using sense. I say you are accurate and you are precise, no problem. All of you are right because anyhow, I make in my mind, I put in my mind that I'm not sure about the last figure. This is certain, say 10.2, okay? Here, these are certain digits. And this is first uncertain. So you are accurate and precise. Another situation, I give them another boy and another balance. Amira told me it is 9.1. Fatima told me it is 9. Salma told me it's 9.2. Are they accurate? No, they are not accurate. Are they precise? Yes, they are precise because using the same balance. But the error here is not a human error, not a human error. The error is instrumental. It is instrumental error. The balance is not calibrated well. The balance not calibrated. There is a pointer here, and the pointer should start from 0. The pointer in this balance started from negative, not positive, right? Below 0. It is an instrument. They are precise, very near to each other, but not accurate. This is the difference between accuracy and precision. In measuring using senses, vision, human senses, but in counting 1, 2, 3, 4, how many students are there in the class? There are 20. They could not be 20.5, of course. In counting, okay, whole numbers. This accuracy and the precision. This is topic number. Look, when you measure fluid in graduated cylinder, especially water makes like this, lemniscus, and you measure here. Measure to the bottom. This is for accuracy and the precision. If you put water in 100 milliliter cylinder, like this, you say, some will say 45, some students will say 45.5, some will say 45, some will say 45.2, and so on. If I magnify, okay, if I put them in another cylinder, calibrated in a more here, say calibration here, 45, 1, 2, 3, 4, 5, 6, 7, 8, 50. Okay, and comes here. So all of us are sure 45. This is certain. All of us will say 45, but some will say 45. Yes, here 45.5. 45.1, 2, 3, 4, 5. Okay, all of us are sure about 45.5. These are certain digits. Some will say 1, 2, 3. The last digit is usually uncertain, and we took into consideration, never depend on the last figure in measurements using senses, as it's called first uncertain. All certain figures and the first uncertain are called significant figures. Significant figures. What's meant by significant figures? These are figures taken into consideration in calculations, right? Another topic or last topic in introduction is studying the microscope, electron microscope, ultracentrifuge, and other. let's postpone these topics to lesson 1.1, 2. Okay, let's complete this in next class. Thank you very much. Thank you, and coming soon.